Thank you very much, and thank you for the invitation. I really enjoyed being here last year. It was a really interesting group of people, both a visit to the labs and the lunch and around, and then the questions were great. And you have an amazing location here, an amazing facility, an amazing group of people. We were just walking around the building, and 1,600 people is about the size of the Stanford School of Engineering. So it's kind of cool to have so many smart people here doing interesting things just down the road. So thanks for the invitation back. When I was here last time, I spoke about the image systems engineering side, and this time, I'm glad to have the opportunity to talk with you about neuroscience. And unlike the image systems engineering involving sensors and memory and image processing and so forth, I'm guessing that not many of you woke up this week and ran an MR scanner anywhere. Is that how many? I'm actually just curious. How many of you have been inside of an MRI scanner? Wow, that's amazing. So it's about, just so you know, it looks about half. I actually also teach the MR physics, the description of the scanners. And so what I decided to do today on the idea that probably most of you haven't built a scanner or run a scanner and so forth, I was going to talk a little bit about the hardware and what's involved in building an MR system, what the physical principles are, a little bit about the human brain, what you might measure using a scanner. There are lots of things you can't measure using a scanner, but there are quite a number of things you can. And then I was going to tell you some stories about things that surprised us over the last 30, 40 years as unusual cases that came across. I'll tell 2 stories like that. And then I will tell you what's kind of driving kind of the industrial application. I'll end with what's driving the industrial application of MR scanners and what it is we think we ought to be able to do if we get our act together. As you know, the world of basic science and neuroscience and the world of engineering, sometimes seem to me like they're on very parallel tracks, that the engineering world is focused on building devices, getting things done, moving them into industry, and the neuroscience world has a big emphasis on basic discovery, which is very important because we know so little, but has maybe underplayed what it is we might turn into an industrial focus for neuroscience. And I will tell you, I think just little green sprouts about what it is you could imagine happening and how that would work out. So that's what today will be about. The first thing I want to start with is just reminding you that the study of the brain and what the brain does is quite remarkable. And even the simplest judgments, how you hear my speech, when you see a picture like this and so forth, your simplest judgments are really massive computations. And that's why, as we were talking about lunch, this notion of the relationship between neuroscience and artificial intelligence, convnets, deep learning, and so forth, has a deep basis because the brain is engaged in so much of this. You can see from this picture here, I just do this, I have 1,000 of these. This is just one of my preferred ones, but there are so many like this. The white chess pieces and the black chess pieces physically are exactly the same. And if I'll prove that to you, I'll drop this thing down, so you come down and slide into place there. Okay, so physically they're exactly the same. And your brain interprets even things like the level of brightness of something, whether it's white or black, is completely an interpretation that your brain makes all the time. And your brain makes interpretations far beyond that. Depth, of course, visual, everything about vision, but interpretations about, well, what does that person think about me? Or how should I be feeling about this? Or does all kinds of judgments and so forth are massive brain computations. And if you say, well, what is it that's the long-term goal? And by long-term, I mean really long-term, like 500-year goal, 1,000-year goal of neuroscience. It's to understand how those computations are implemented. And hopefully on the way, before we get there, we will find interventions to make people who have trouble seeing or trouble reading or trouble learning or trouble remembering or trouble having a stable emotional state, we'll find ways to be helpful as we uncover more and more about the way the brain makes judgments of this sort. So what are the parts of the brain that we can get at with magnetic resonance imaging? Well, these brain computations that are so important to us depend on a collection of different cell types. The most important and widely referred to is the neuron, the nerve cell. They're in a very important class of cells. In the human brain, the nerve cells are largely in the neocortex, are contained in this sheet that covers, this is about a 2 to 4 millimeter thick sheet of nerve cells over here. And their cell bodies are located over here. And the neuron is the part of the cell that is the cell that sends out action potentials, little brief electrical pulses that perhaps I'm guessing you're familiar with. When it sends those pulses out, some of them are just local and some of them go over longer distances. And the ones that go over longer distances travel on something called an axon, which I'll show you in the next slide. So axons, so the nerve cells are here in this outer region called the cortex. Here the brain, half of the brain has been cut away, and you can see the long wires that connect up different parts of the brain over here. And these long wires is called the white matter of the brain. So the structures I'm showing you right now, kind of this sheet of cortical neurons about 2 to 4 millimeters thick, and the axons, or the long connections that connect different parts of it, those are at a spatial scale that you can assess their health and activity and structure using magnetic resonance imaging. And that's why I'm focusing on that here. I would say there's one other thing about these connections that I think you can imagine happening in, maybe it even does in some ways in the world of electronics, which is that these are the communication channels, and for a long time people thought these communication channels were kind of fixed. You'd grow up, you'd get to be about 5, 6, 7 years old, and these communication channels, they were there. They were your white matter. Nobody really thought much about it. But over the last really no more than 8 to 10 years, people have come to realize that these white matter connections in the human brain are themselves changing when you learn something new. When you develop a new skill, the white matter tissue properties actually change. So these aren't passive wires like the wires that come out for the building here. These are wires that actually, when they're used more and more, they kind of change their properties to get better and better at carrying signals in certain ways. And we don't really understand that very well. But it's a kind of one of those high-level architectural differences that you wonder whether there might not be some application for that in the design of system-wide electronics. The other thing I wanted to get you started on thinking about is many of you will pick up a magazine or a newspaper and see a story about neuroscience discovery. And to those of us on the inside, who, I spent a lot of my life thinking about neuroscience, it's not, you can read a whole New York Times article and not realize until you get to the end that this was about a worm, or it was about a fly, or it was about a mouse or something. And I think that's actually important, and it's something I'm trying to, whenever I find myself a reporter, I try to get them to be clearer about this. The ability to translate between different systems is a real major challenge for neuroscience. It's a really significant factor. And these are pictures of the human brain, the closest primate, the macaque, not the closest, but a close primate, the macaque is shown here. A mouse brain is shown there, and you can see that the volume is 3001. And the notion that you would be able to learn everything you need to know about the human brain from studies of the mouse brain seems to me a stretch. And yet, it is an important feature of the way neuroscience is largely conducted worldwide. And there are many problems with that, and one of the things that I push, as you can tell, I'm not shy about this, and one of the things that I push is that the notion that we have to understand when it is you can translate between these systems, and when there's something you want to know about the human, is it okay to use a mouse model for that? And I think getting clearer about that is an important thing, and something you might look for when you do your readings and think about these things. So as I showed you before, the brain volume, typically about 50, well, this is just in volume as opposed to weight. It was in grams before. but this is the typical brain volume of a human. There are specializations throughout the brain. These are just kind of geography. Front, occipital is the back, parietal is over here, temporal is over here. This is the cerebellum. It's another important structure. It's got does a lot of important things. And our task is to understand all of these parts, the connection between them and what they do. And at first, we kind of make rough assignments. So for example, visual cortex, I'm telling you this now because I'll use this a lot of the responses to visual stimuli are initiated back here in the occipital lobe. They're used by the other lobes, but they are initiated. We learn a lot about vision from studying over here. There's a part of the temporal lobe over here that is very important for hearing, speech, language, and so forth. People often think of thought, memories, moods, emotions, and so forth as being associated with other parts of the brain. And I show you a picture like that at that resolution, and then I really want to remind you that the neuroscience field broadly traverses an enormous range of scales. I was showing you pictures. These happen to be a picture I had of the white matter pathways that we've assessed using magnetic resonance imaging, and these are the ones that stay at the back of the brain, and these are the ones that travel somewhere else. And that's the scale we were just talking about. But I have colleagues who will take out a little piece of a brain like this. And this is a 100-micron scale bar, study a stack of neurons like this. I have colleagues who will take out a little piece like this, blow it up over here. This is a 10-micron scale bar. And I have colleagues who will take out a little piece like this and study this part, and it's a 500 nanometer scale bar over there. So that the scales at which people will talk to you about the nervous system are just extraordinary, right? It's like talking about wiring up the whole building versus telling you about a transistor inside of one component here. And in engineering, we're not great about traversing all the different scales. It's not as though there's never any pushing and shoving between the transistor folks and the systems level folks. That's there plenty. But let me say that in neuroscience, it's far behind even that. We don't really have even a good push for building models that go from the scale of hundreds of nanometers to the scale of the whole behavior or the activity in the whole brain. And that's something that, particularly at Stanford now, we're just putting up a Neurosciences Institute building. We've taken a whole chunk of it, core and the center core, and devoting it to people who will work on theory. And we're hiring 6 theorists to come in to think about these things. And one of our major goals is to help develop an engineering/science of how it is you can traverse different scales so that people at different scales might work together and help one another when they make these discoveries. And I can tell you right now, you will probably be at a talk sometime in the next, or read a newspaper article sometime in the next 6 months or a year, and somebody will say something about a molecule and tell you something about behavior. But let me just alert you that traversing those scales is not something we're all very good at yet. Okay, so now something about the instrument itself, which will operate at a particular scale. It'll operate at the scale of a couple of millimeters, basically, for mostly what we do, though it's a complex story, but let's just say it's going to be a relatively coarse scale. So this is what an MR scanner looks like. Probably looked a little different from the ones you went to. This is a research scanner. When you get into one of these scanners, it's quite phenomenal that you can do anything at all. They're non-invasive, right? So you don't have to crack open the skull. You don't get poked with anything. You just kind of lay in there. And we're going to manipulate the magnetic fields in a way to make inferences about the many kinds of inferences. That's one of the fantastic things about MR, is you can draw all kinds of inferences about the structure, the activity, the connections, the molecular composition of the brain. The scanner comprises, well, here's, you get on a bed, you get put into the bore of this fixed magnet. So here's the big iron over here, and it's got a solenoid wrapped around it, lots and lots of wraps of solenoid around it. And the wires that are around it are superconducting, so that they're kept very, very cold by liquid helium. And you inject some current into these wires when you first turn on the magnet, and it just goes forever. You just keep it cold, and it's got zero resistance, so it just keeps this iron magnetized. And the typical levels will be somewhere between 1.5 Tesla to, at the ultra high field, you'll see things at 9.4 Tesla, 11 Tesla, things of this nature. And that's a steady magnet. And then to make images and be able to make inferences, there's a couple of other parts. We can modulate. So the magnet produces a main magnetic field shown here. We modulate this magnetic field through the use of what's called gradient coils. We also apply little radio frequency pulses to your brain to evoke activity, and I'll show you this in a moment. And it's the use of these radio frequency coils and gradient coils in combination that's called pulse sequence programming. And it really is a whole programming system that you use to try to learn different things. And people are still being very creative about the ways in which magnetic resonance systems can be programmed to learn different things about the brain. The fundamental physics, and of course in a talk like this I can't do a lot on this, but the fundamental physics is if you imagine this beaker is like a brain, or one voxel in the brain is filled with water, and you put it in this steady magnetic field, when you apply a radio frequency pulse over here, and that's what's shown, the pulse is being applied here, and then you listen after you apply the pulse, you will get a radio frequency pulse back out. And that pulse will depend on things like the field strength, so therefore these gradient coils matter. It will depend on the tissue, the nature of the tissue. It'll depend on a number of things that you might care about. And the basic science of magnetic resonance was to figure out how to apply these pulses and manipulate the gradients in order to learn about this. And just as an example, if you have two beakers over here and you apply one of these pulses, they'll both come out with the same MR signal. But if you put a little gradient across these beakers, so this is in a lower magnetic field and this is in a higher, by putting a little gradient across them, the actual frequency that comes out will change. And so you can use information like that to localize where the signal is from this place or from that place. And again, it's a 10-week course to go through all the ways in which you would do that and make the beautiful images that people can make. But simple phenomena like this lead to the industry of magnetic resonance imaging. Okay, so I've been focused almost entirely on the part right over here. The subject gets in over here and so forth. But there's control rigs for these gradient amplifiers, for controlling the radio frequency pulses, for generating waveforms, for keeping the liquid helium flowing around here for the system, for reading the data out, for processing it, so forth. So that in fact magnetic resonance instruments comprise a system that costs about $1 million a Tesla, basically. So it's about $3 to $7 million, depending on the one that you get. And there's a couple of main vendors on this, and there's also GE, Siemens, Philips are the big ones, and Bruker has dominated the market for things like mouse imaging and so forth, where it's also being applied. Okay, so what do you measure? How do you measure brain activity with one of these things? Well, we don't measure activity directly, but here's the way we can get a sense of activity. And this was only really worked out a bit and discovered from around 1990. So we're talking about a relatively new discovery here. And the first demonstrations of this in the high-profile journals were in 1992. So this isn't old stuff, but the basic idea is illustrated by this story. Fulton was a famous neurologist working from the 20s through the 50s. And Fulton, at one point, as part of the story, he had Walter Kay come to his clinic. And Walter Kay had never been able to see very well on the right side of the world. And it's because he had a lesion in the left back of his brain over here. And Walter Kay says, Doc, I never could see very well, but it really hurts me now. It really feels, that's like I'm in pain. Could you do something for me? So in 1928, what can you do, right? You don't have MR scanners and so forth. So what you did, the standard of care back in those days was to crack the guy's skull open and have a look on the inside. It's a surgery. because he's in pain. He can't actually do anything. And you don't know what's inside his head. So you break open his skull and you take a look. And that's what he reported here. So on turning down a left occipital, back of the brain over here, occipital bone flap, a large angry-looking angioma. Doctors always write this. They still write this way in emotional terms about what it is that they're looking at, was disclosed. And it extensively involved, as you know, occipital, the visual cortex. Now the hemorrhage, the bleeding, occasioned by the bone flap was so excessive, right? You open this thing up, oh my God, it's just blood everywhere. Close it up. So we decided the operation had to be abandoned without touching all the invasive growths in there. And however, a decompression was made. So there was less pressure on the guy's skull, and he felt better. His vision was a little bit better, and he felt better. Okay, so this guy, Walter K, is happy. Now, in 1928, when you can put the skull back on, they couldn't put it on as tightly sealed as we do now. So it was good, it was on, but it was a little crack. And Walter K comes back to the clinic a few months later. And when he comes back to the clinic, he says, you know, Doc, it's weird when I'm out there. He was a sailor. When I'm out there on the boat and I try to see something in a distance, my mates tell me that they can hear psh, 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 coming right out of my head. What do you think that is? So Fulton, a smart guy, said, I don't know, it's probably the blood flowing there. So he says, listen, instead of looking carefully, count backwards from 100. Nothing. Okay, so he's thinking and nothing. Okay, listen to my watch ticking. Nothing. Okay, look at that little thing. Psh, 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 and he records it with a stethoscope, and he says, so what did he learn from that? He says, you know, the blood flows to the part of the brain that's active, and if you do something else that involves the frontal cortex or the temporal cortex, that, you know, there's no blood flowing back to visual cortex. But if you do a visual task, you know, you can hear the blood there. So that the blood flows to the active parts of the brain that we're using, that's the principle, and that's what we can pick up in the MR scanner. So we take somebody like Walter K, or you or me, and put them in an MR scanner, and we have methods for detecting where there's more blood oxygen and where there's less blood oxygen. And that's because the magnetic field properties of oxygenated blood differ from the magnetic field properties of deoxygenated blood. And so this signal that we measure is called the blood oxygen level dependent signal, and that's what we can measure. Now you should know, and this is just I'm now just telling about a little fight I'm picking with my pals, that because this is such an indirect measure of neural activity, functional magnetic resonance imaging, the ability to measure this, is often dismissed by real neuroscientists who say, well, that's just an indirect measure of neural activity. I stick in an electrode and I measure the actual electrical activity, and that's the thing. And I just find that peculiar. And it's a sense in which one would dismiss the LIGO measurements of measuring properties of the origins of the universe, because those are indirect measurements of the origins of the universe, as opposed to what? You know, you take what you can get, or that I'm looking at you and you're seeing, or I'm looking at you and seeing the colors of your hair and faces and clothes and so forth. Those are indirect measurements also. The photons are coming to me and I'm seeing the scattered photons. I'm not touching and measuring your shirt directly. So indirect measurements can be very useful. So that's just me getting over it. Okay. So there has been remarkable progress since that discovery in the early 1990s. This is what brain images used to look like using positron emission tomography. This is from a Nature paper in 1986. And at this point in time, the voxel sizes have shrunk massively. Our ability to see brains and brain structures, identify them, so forth, the resolution has become massively better through the persistent application of these technologies. And amongst the things that we can do is we can, now without hurting anybody, we can do an MR scan and find segment out the gray matter. We can have them look at something, and we can see the activity as if you're focused here and this signal moves on out to the periphery like this, you can see a traveling wave of activity that shows you that this piece of the brain in your visual cortex actually represents the visual field from fovea from the central visual field to the peripheral visual field, from the central visual field to the peripheral visual field in an experiment that takes 10 minutes. So by the way, everything I'm telling you today is published somewhere. That particular observation we published 25 years ago. Okay, so when the graphics weren't as nice, but that was one of the first things that we looked at for a while ago. So you can see how it is the visual field forms a map from fovea to periphery in the back of your brain. Notice this red spot here, that was news. We didn't know that there was not only this central representation over here, red meaning the center, and green, blue, purple showing the map, but there's another representation over here of the fovea. And actually it turns out that we now know 5 of them, and that was new discovery that was about the human brain. You can map the other dimensions, some of you may be wondering, rotate it around this way, and you can see that these actually separate out. There's not one simple map, but actually these maps, there's actually, we know at least 20 maps that have been replicated by different labs at this point. And these maps, from these two kinds of measurements, and these maps, kind of tile the back of the brain over here, and different people, almost everybody has the same maps, but when somebody can have a problem with vision of one sort or another, they can have a defect in one map or another. And so this is giving us kind of a baseline of data of what we know about the brain. And one of the things that's true is this is pretty universal. It's not like I'll look at folks over here in different places and they'll have different maps. You'll all have these maps. They may be a little bit different in size, but they'll all be in about the same brain. And so this is part of the genetic program of the brain. And this is all new information that was picked up over the last number of years. I would say this tiling of the maps has been pretty much settled by about 2005 to 2010, not that long ago, but there it is. So the next evolution, the next stage in this processing is to start building engineering-like models about the responses here. And one of the things that is sort of classic in vision, if you can do it with a microelectrode with a single cell, is you can put in measure that cell and ask where in the visual field would you provoke a response to this cell. So if you put this, you just saw that in a way. If you put a stimulus in the middle or in the side or upper or lower, would it bring out a response? And that's called the receptive field of that cell. And you can do the same kinds of measurements in the human brain with an MR scanner. You can measure a particular location. We sometimes call this a voxel, a volume element. Measure a particular location and ask what parts of the visual field will give rise to a response over there. And you can do that literally by just putting on a stimulus, like one of these stimuli that move around, and here's a measured bold response, observed, is shown over here. And if you say, well, my theory is that the receptive field is here and it's about this big and so forth, that would make a prediction. And that prediction's way off, so how about if we move it over here? Okay, now it's a little bit better. How about if we make it a little bit bigger still? Now it's better still. And you go and build engineering models of how each voxel responds to stimuli in different parts of the visual field. And this program of research is getting bigger and bigger and more solid. We now have kind of standard cortical models of the visual responses for certain classes of stimuli. And we fail on other classes of stimuli, but that's a program of research that a number of people have really adopted. And we're getting quite quantitative data about the sizes of receptive fields. Over here, they tend to be small. On the lateral surface of the brain, they tend to be big within each of the maps. And it's turning into something that'll seem more familiar to you folks as engineering curves that characterize the standard performance, standard structures and sizes and properties of these different maps. And once you get that, people start to take these standards and apply them in all kinds of other contexts, and that's what's reviewed here in this paper. People look at developmental plasticity, what happens in aging, what happens in the autistic brain, what happens to these things when people have Alzheimer's and so forth. Excuse me. But the development of the basic engineering standards is something that's required, I think, to make good progress in how it is you could apply these in an industrial setting. Excuse me. Okay. So these are papers illustrating that. So now let me tell you a couple of stories. It'll really be 3 stories, and I'll keep going at this pace. Do feel free to stop me if you need to, but I'm going to go through this. The first is, I'm sitting in my lab minding my own business one day, and this friend of mine who's an ophthalmologist calls up and says, we've got this person who showed up in the lab who has a strange white matter set of connections. And this is the normal connections. Here, you've got 2 eyes. The stuff near your nose over here that comes out of this eye goes to the other hemisphere. Near your nose on this side goes to the other hemisphere. The stuff that's on the side of your head stays in that same hemisphere. And for that reason, you end up with the right side of your visual field goes into your left brain, and the left side of your visual field goes into your right brain. And that's true for all of you. There's nobody in this room for which that's not true. Okay, and that's just shown here. Okay, the optic nerve from each eye meets. This is called the chiasm. Half the fibers cross, and you end up with the left visual field comes over. You're looking at the brain here from the bottom, so that's why this is the right brain. Okay, so this is what a normal brain looks like, and we're pretty good at tracking the fibers at this point. This is another MR technique that I'm not going to explain to you the physics of it or the algorithms, something I've spent a lot of my life on, but we're pretty good at this point at tracking these fibers and how it comes out from here and back to the brain. Here's this person. Something seemed funny. You don't need a degree in radiology to see that, wow, okay, like there's a gap there. Where'd the chiasm go? So this is a person who showed up because he was 24 years old, I think, when he showed up, and his eyes were moving around in an unfortunate way, which happens to lots of people, by the way, not only to this person. His eyes were moving around, unfortunately, but he had no chiasm. So that means this person's visual system must have been really different. And they were a little shaky about this. Could you look at this guy and who has this kind of mapping, where each eye goes directly here to the corresponding size, not the half eye. And we could confirm that. No, this is the ophthalmologist measurement and decision. We could confirm that by putting him in the scanner just functionally, blocking off this eye, and we put something in this side of the visual field, and the left visual field stimulus actually went into the left hemisphere, which never happens. It's also the case that if we put it in this side of the visual field, comes into this eye, it drives the same piece of cortex. This person is not at all like you or me, and we could go build these models, these little receptive field models of the responses in his brain. And if you do the usual thing for you or me, you'd have any voxel would have a response, would have a receptive field like this, and you could predict everything. You'd see, wow, we really missed a lot on this guy. But if we built a model like this, where it spanned symmetrically the 2 sides of his field, you could see we could hit everything. We could get the whole thing fit. So this guy grew up his whole life with, instead of having just half of the retina project in over here, both sides of the retina, this side and this side, projected in to the back of his brain in an interleaved way that gave him these very unusual receptive fields. And he could see fine. He played baseball. Right? He climbed walls. One of that rock climbing wall, interior wall kind of thing. He could read just fine. And so I don't exactly know what to tell you about this, other than to say that despite profoundly disrupted visual cortex, V1 maps, I mean, these are not like anything that you would see. This guy, the rest of the brain figured out what to do. So at certain point in time, the rest of your brain can quite plastically adapt to changes in the input that are completely unexpected. And that's an important lesson. I can tell you that if it happened to me at my age, the rest of my brain would not adapt. And I can tell you that because of the story of Mike May. There's another guy, I worked with Mike, a number of us have worked with Mike over the years. Mike had a slightly different, so the first guy I told you about, that was at birth. Right, that didn't happen by an accident or anything. At birth, he had no chiasm. Mike, when he was 3 years old, was in the garage of his parents' place, and they had, I don't quite know what it was, but there were some chemicals laying about, and he knocked it over, and there was an explosion, and one eye was completely blown out. Now, 3, remember, you can see just fine. 3-year-olds have very fine vision, and their brains aren't all developed, but they can see just fine. But he lost one eye, and then the cornea and the lens in the other eye were very severely damaged, so much so that they couldn't put in a new cornea and lens for him because there were stem cells that at the time, he was 47, he was 3, this must have been around 1990, maybe 85 when this happened, they couldn't do, the stem cells could not be replaced at that time. But when he was 47, it became possible, because of advances in stem cell technology, to put in a new cornea and lens that would stay and not cloud up right away. So the question then is, what happens to somebody who, for the first 3 years of life, the brain is developed and grown in a certain way, then loses all contrast vision in the one remaining eye, and spends the next 44 years without any contrast vision, and now we can repair it, what will happen to that person's vision? And we had some examples from the literature. This was a guy who got one of the first corneal transplants in England in the 1990s. And let me not tell you the story, I'm worried about time. And what it is that happened is that he went from being a blind man who was very successful and had his own shop and so forth. And when they did the corneal transplants for him, he looked at the world, and he would look at something like this, and he might touch it and kind of understand what it is. But then if he moved to here, he had to sort of figure it out all over again. Whereas you could see it as an integrated object from your experience and recognize it. For this fellow, nothing like that. He was always reasoning about the visual signals that came in. So he would see a bus over there, and then all of a sudden the bus would be here, and he wouldn't see the continuous flow and be able to judge the size of its motion and distance and so forth. He became terrified. He used to stride around the streets of London with his white cane confidently. When he had his vision restored, he became terrified of lots of things. He actually died a year after. The suicide rate from these miracle recoveries is way too high. Okay, so this is from an analysis from Gregory. We did these same kinds of experiments with Mike, and the first thing that worried me when I met Mike was, you know that these don't often go very well. But Mike is quite a character, quite a guy. He's a very confident guy, and in fact, so I've told you his story a bit. Mike said, he was going to be fine. In fact, Mike is winner of the gold medal in the Paralympics for skiing downhill. And the speed is, he could ski downhill at 60 miles an hour. And you might be wondering how it is he could do that. Well, the way you do it is you get the gold medal winner of the other Olympics to ski in front of you and tell you, go right, go left, jump. Okay? So he's an exceptionally courageous guy. There's a book about Mike called Crashing Through. And so he is that kind of adventure seeker. And he went, and here he is as his cornea and lens was first replaced when they open up his eye, and the woman who's blonde hair, you can see there had been his wife already for 10 years. And there it is, he's first looking at her. And it's kind of heartbreaking to see this. She's saying, oh, I hope he likes the way I look and stuff like that. And she's very sweet, and he's a very, very nice guy. In any event, over time, we measured Mike's spatial sensitivity. What's the finest pattern? Higher and higher means finer and finer patterns. Here it is, low patterns, high spatial frequency, that Mike could see. And over a period of 21 months following the surgery, he really never became, this is what normal, typical controls see, this is what Mike could see. I happened to go by Davis, where he was living some time later, and picked up one more measurement, and he's still way off the distribution. So now we learned from Mike, in this particular case, that his vision, after having been cooked for 3 years in the normal development process, did not develop correctly and properly after this miracle cure. And because we now have magnetic resonance imaging, unlike the Gregory case, which was very important, we could add to that case in the historical documents things about brain responses. And you've seen these maps of red to green to blue and so forth in the typical brain. And in Mike's case, all the really small central stuff was kind of missing. And it had lots of imperfections there. It was as if the small cells in his brain that carried information about the fovea were lost because they were never excited after 3 years of birth. And when it got fixed, when he was 47, even it's now been, I think, 10 years since the surgery, there's been no real improvement. The one part that did respond quite well in Mike is a part on the lateral side over here, which is motion selective. And Mike skied a lot, he moved around a lot, and he could tell lights flashing on and off, and that part seemed like it was like it continued. So there's Mike skiing. There are parts in your brain and my brain that respond particularly to faces or objects, and that's shown over here. But in Mike's brain, we really could never evoke any responses to those kinds of things. So again, this is just in the storytelling mode for how many. What do you think that means? Okay, I have about 10 more minutes. Are we okay with that? Okay. So what I'd like to say about Mike and about these two stories is that there are certain moments in the lifespan where, wow, your brain is really flexible. You're born without a chiasm, go ahead, it'll work out, okay? But then there are other parts when you're 3 years old, or when you're 3 years old and things have gotten started in a certain direction, and if you muck up things at that point, it's almost impossible to overcome unless you deal with it while the child is still young. And there's a big debate in the literature about how much plasticity there is. Is the brain plastic in the adult or not plastic and so forth? And that's fine. People enjoy that debate. But let me say the answer is really obvious once you spend a little time on it, which is at some points in time, some parts of the brain are plastic. At other points in time, they're not plastic. And our mission really as neuroscientists is not to decide, is the adult brain plastic? Sure, parts of it are plastic. Parts of it aren't plastic. Our mission is to define the engineering curves and figure out which, when, let's get serious, under what conditions and so forth. And that's, I think these stories feed into that, and that's my story, and I'm sticking to it. Okay, so the last bit then, the last few minutes, I'm going to race through. Well, okay, so great. So you can do these things, they kind of get published in journals and so forth. But suppose you wanted to deal with, some application of vision. And an application of vision I'm quite interested in is a child comes to you and has difficulty reading. And reading has something to do with vision, right? You need to see the text on the paper. It needs to go into the rest of your brain. It has something to do with language areas. Because the text has to be communicated to language areas. As it turns out, sounds and speech are very important for learning to read also. So it's kind of a whole brain thing. I like working on reading for another selfish reason, which is that none of my colleagues who study mice compete with me on reading. Okay, they don't look for the mouse model of reading. So in the case of reading, you can imagine the text starting out in primary visual cortex, back here in occipital lobe, and then traveling its way through. It turns out that there's a particular spot at the bottom of the brain that literally has been given the name, the visual word form area, that when we read stuff, the signals always evoke a response over here in this VWFA, the visual word form area. Other responses too, you can see the responses. Again, here's primary visual cortex, a word, you're fixating here, a word is presented here, you see the response here, you see the response here, you see the response here, you see it in this region that appears to be specialized for words. So, you can kind of track the signal as it goes through the brain. And when you get to this region over here, you might wonder, well, how could I characterize this child? What could I do as somebody comes to you with a child who's not reading well? What's the standard response that you would see in this part of the brain of a typical good reader, and how does this particular child compare to that standard response? And there's different ways in which to measure it. I would say I'm now taking you to the edge of the field of where we're not sure how this is going to go. But one thing you might measure is here's that region down here, the visual word form area. You might say, how much of the visual field does it respond to? And one of the things we've just learned, and now I'm telling you like 2-3 years ago, we've just learned is that when you get to this part of the brain, it really, this is the visual field, here's the center. So this is a very small, your whole visual field is about plus or minus 130, plus or minus a total of, let's say, 130 degrees of visual angle. So what will drive this will be only the little bit over here, just the central 8 to 10 degrees. That's all that ever gets in there. So when you put text out here, try it yourself sometime. You know, when you put text way out here, it's impossible to read for a variety of reasons. Some being resolution, some being the reading circuitry just never gets a shot at it. So we can look at how much of the visual field gets into the reading circuitry in this child's brain. And so the reading circuitry is shown down here. You can look around at the other parts, and there's a lot of it that gets in there. It's much bigger. But the reading circuitry only sees a little bit towards the center. And then you can look at that in child to child to child, different children. And actually, that differs quite a bit. And so that's the first thing that surprised us. And so I will happy to talk to you about why I think that would be and so forth. We're suspecting that we will look at these children and their eye movement patterns, and their eye movement patterns will interact with how much of the visual field gets in there. But I wanted to give you a sense that one can start to do little bits of measurements of typical children, and then I will show you later children who have poor reading. The second thing is, these wires, these connections can differ from person to person. And you can go and, some of them are very big and some of them are more localized. And so the question is how do these connections fit in the circuit? And we were capable of measuring the properties of some of the major wires over here, how they change in a particular tissue property. I'm sorry, I'm not explaining to you, but it has to do with basically how oriented all the fibers are in a particular regions of the brain. And in some kids, as they got older over the 4 years of the study, the tissue property would increase, this measure would increase, and in some they would decrease. And it tended to be the case that the kids who were poor readers, it decreased, and the kids who were good readers, the blue ones, tended to increase. And these are ordered by increasing versus decreasing. So this again has to do with that point I made at the very beginning, that these fibers in your brain are active and respond to the needs of the system. And so another place where there might be an issue when you try to diagnose kids is the tissue properties of these white matter fibers. In fact, this effect, when you average across groups, is actually quite big, but let me not belabor that. So the place where we're failing, and this is how far we are at this point, and this I'm close to the end now, is when you look at those slopes, where you're getting bigger over here or where you're getting smaller, you can see that there's a correlation. The ones that are the FA is going up over time, the ones where the FA is going down over time tend to be poor readers when it's going down and better readers when it's going up. And you explain some fraction of the variance, and this is good enough to get you a paper published in a journal. Okay? But it's not actually good enough. And you can see why, because if you come in here and you look at the spread in reading scores given some slope, it's just way too large. It's not helpful to the parent or the child. Is this really a problem for you, or is it not a problem for you, and so forth? And so the cutting edge of the field is to get better measures, understand more parts, how they work together as a system, and to try to give better advice to people about what's likely to work for them or not. And that's what we're engaged in now. That's kind of the mission. The spread, this correlation of 0.51, that's, you're only explaining 25% of the variance. You can actually go from, try to predict a reading score and measure the kid's reading score, and you do okay, and it kind of looks impressive, but it's not good enough for diagnostic work by far. And so doing one track at a time versus 10 tracks, doing other measurements and relating them seems important. And so I would say that where the field is now, after all these sort of single case studies and so forth, is can we build up an industry in which we measure multiple tracks, measure multiple ways with these with these kinds of visual field coverages and the tracks, and large scale in children where we have reading measures and start to build a more precise characterization of the healthy child and which parts are out of spec for this child that's not learning to read. And for some of those children, we can do something. And for some of them, we need to give them audiobooks, or we need to teach them in other ways and so forth. And people, as you know, I don't know if you know this, but people can be very successful in lives. Like Charles Schwab can't read worth a damn, okay? And he's had a pretty successful life. So, you know, you can be very successful in life given the opportunities, even if you can't read. It's better if you can read. It's not like Charles Schwab doesn't want to read. He thinks it would be better. But you can still do quite well. So we need to find ways to diagnose which are in, which are out, which one can help this way, that way, and so forth. And reading is an example, and I chose it for the reasons I told you. Reading is an example, but actually this is an instance of a very large set of clinical, psychiatric, neurological disorders that could involve mood, that could involve anxiety, that could involve all kinds of things, where again, if we could work it out in the case where we understand vision and reading is kind of a controlled place, if we can do well there, we might set up the infrastructure for being able to do well in lots of other cases. And I feel like we've done in the vision side, and it's usually the case in these neuroscience, psychological things, that the senses are the first places where you work things out, and that they kind of slowly migrate into other areas. That we've done okay with finding the maps and developing these computational receptive field measurements and following them down into here and adding these things. But there's just so much more to be done, and there's just it's great opportunities for people and great potential, and it'd be. Thank you very much for the opportunity to come here and tell you these stories.
Thank you very much for the presentation. I got a specific question. My son participated in an MRI study for autism. What have you learned? What have we learned so far?
So let me be specific for you. The group that is doing what I consider the most advanced work on autism is the Simons Foundation for Autism Research. They're located in New York. And we've helped them collect data and analyze brain imaging data. There's a second group that's also very good up at UC Davis at I think they call it the Mind Institute. And there's a set of studies across the scale from mouse models of autism to brain imaging of autism and so forth. And I would say that what has not been what has been discovered is various little tissue differences and size differences and developmental trajectory differences from the growing, as the brain grows, what grows first here and first there, and the timing seems a little bit different in certain kids with autism. What the big thing I would ask for is, and I'll get shoulder to shoulder with you on this, is a lot of kids get the autism diagnosis, but just like the story I'm telling you here, the reason for the autism could be quite different. And therefore, the way in which you want to help the child do the best that they can could be quite different. And the number of classifications that have been agreed to by the autism community from MRI is not yet solid. And there are various little proposals. The group at Davis will say one thing, the group at San Diego will say something else. And a lot of people just go straight to the gene. And so if I'm giving you a sense of confusion, some of that is mine. Some of that is just the state of the field. And I would say for the very latest, I participated in this kind of work up till about 5 years ago. For the very latest, I would click on the Safari, Simons Foundation for Autism Research website. They're pretty focused on genes. For whatever reason, people are focused on genes rather than the global. There's a smaller group that do the MR things.
Okay, so I have a softer question and a harder question. Softer question. Well, I was told that the reason why MRI takes so long, it's like 40 minutes, 1 hour, is because of the software. It's really slow in processing the image and building. That was kind of 10 years ago. So there has been any progress in that, or do you see opportunities?
Yes,
for software?
I'd say that's not true at this point. That at this point, the reconstruction, so that phase where you go, the signals come out of the scanner, those oscillatory signals come out of the scanner, and then the time between that and an image is real time.
Oh.
So before the scan is even done, you can get a look at the images for most types of images. All the ones I showed you today, you can get a look basically in real time.
Okay. The harder question is, that thing is really noisy. So do you see that like the part of the brain that has to do with sound is always over? and if you were to study that, how would you go about it?
So, you know, whenever we put anyone in there, we put them in with earplugs. Yeah, and let me tell you, the source of that noise is, remember I told you about these gradient coils that change the mean magnetic field a little bit? So they're sitting in there, and there's a lot of force. When they change the magnetic field a little bit, there's a lot of force on the literal piece of hardware that's there, so that they're held in place by very tight screws, but they're straining against the screws, and that produces this noise, and you're hearing them go back and forth as they change the magnetic field. And people have invented silent pulse sequences, so that greatly reduce the noise in the sequence, but then extends the time and lowers the quality. So those have been patented. Companies have started based on this. We don't use them at our place. We just put noise canceling devices or noise blocking devices in the ear. So people have looked at that, and other people are bugged just like you are. But, you know.
Hi, thanks for your talk.
Thank you.
I just wanted to ask, for patients with mental disorders, have you seen any correlation of using MRI as a therapy to help them?
No, I've heard of it and never seen a case of it. I have heard of groups who claim that putting people into the MR scanner and modulating their brain with the magnetic fields has a beneficial effect. It's not something I have firsthand knowledge of or can confirm, but I have only that I've heard of it.
Okay, thank you.
Hi, I have a question. You focused on the reading aspect, but have you all done any studies on hypersensitivity to hearing? Any hearing? I know the machine bangs around, but I was...
I have not, but there are quite a number of groups around the world who study auditory cortex, which has its own set of maps.
Okay.
And
where would you find information?
Yeah. You know, if you send me something, I hang out with people in otolaryngology and audiology, some, and I'll ask them for... I'm Wandell at Stanford.edu.
Hi, I was just wondering about so many MRI, but every time they inject some contrast fluid, so what difference does it make?
Yeah.
With or without contrast? What's
Yeah,
the reason?
you know, the goal is to avoid, we try to improve pulse sequences so that we don't have to inject contrast, but to measure the distinctions between certain types of tissues and certain pieces of the brain, people will inject contrast to get a higher signal to noise ratio. Sometimes they'll inject contrast and tag it to certain molecules that they're in, you know, have it go to certain molecules. And that's the goal, is to get a higher difference between whatever the contrast is labeling versus stuff around it. And you know, it'll all go into, say, this organ, and then that organ will glow and you'll be able to see its shape and so forth, or but that's what the contrast agents are for. At our center, which is a basic research, and we do it on undergrads and so forth, we never inject contrast agents, but they're there for various purposes, particularly for clinical diagnoses.
Thank you.
That was one of my questions, but the second question is, how far do you think we are close to open MRI or mobile MRI technology that
Yeah, we're both closer and further. When I first showed up at Stanford, we had an open. It might still be there. We had an open MRI for surgery, and it was kind of a half. I think it was a pair of donuts, and the surgeons all had MR compatible devices, a low field strength, and the patient was in there, and they would do their surgery and be getting updated MRs at the time. And then they just stopped. It just didn't work very well. I'm not sure why, but they stopped. And it was kind of low demand for those particular instruments. So it was there, it went away, and I've heard that people now have new designs that they think might make it cost efficient. The guy who's worked on the kind of basically portable MRI, I saw give a great talk. It's a guy named Larry Wald, W-A-L-D, at Mass General, and Larry's kind of, for many of us, he's the guy who you want some MR hardware design. He can design anything, and his goal was to put something in a suitcase and carry it to Africa around to clinics for a diagnosis, and he had various ideas about how to do this and so forth. And I don't know, the last I heard his talk, it was, wasn't ready for a company, but it was about 3 years ago. And I don't know to what extent he's still working on that. And I see him for other reasons. I haven't asked him.
Yeah, I think it would be great for like sports injuries. People play a lot.
Yeah.
I read an article in the
the Third World was the one that really had him driven. He was describing these situations where you go to certain things you could diagnose in the clinics in Nigeria and Africa and so forth that were just impossible to diagnose. Without an MR, and he was quite motivated. And it seemed like great if you could get it done.
Thank you.
Thanks.
The last question.
Thanks for a nice talk. One general question I have is that you talked about correlating mice brain studies to human brain studies. So I'm just wondering that is there any functional difference in mice brain cells versus human brain cells in terms of actual biology, like neurons or axons or frontal lobe and certain brain structure, or they are essentially same?
There are a huge number of differences. There are, but there are a huge number of similarities. So amongst the similarities is if you look at the basic mechanisms of the action potential, the channels and the nature of the transmitters that communicate between cells, they often are very similar, and not always identical, but often very similar. So there's a lot of good reasons to study certain things in the mouse. If you look at other things, such as the number of different types of cells. So when you look at, if you don't just say neurons, but you say neurons of this sort, you might not even find any like the ones of interest in the human in the mouse itself. So neurons have a characteristic shape, set of pattern of connections, and so forth. And you can't always even find that type of neuron in the mouse itself. I could go on. The pattern of connections, it's 3,000 to 1 in volume. The pattern of connections, in limited cases, you can say it's kind of the same, but the mouse visual cortex is completely different, laid out if you want to say, well, I can sort of see some vague similarity, okay, but you'd never confuse them in a million years.
Okay, thank you.